Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we will welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, John Zmuda from Thermo Fisher Scientific.
Thank you. As mentioned, this is Jonathan Zmuda, Director of Cell Biology, Thermo Fisher Scientific at our Frederick, Maryland site. And today I'll be talking to you about enabling large-scale manufacturing of viral vectors in the CTS LVMAX Lentiviral production system. So in the recent years, the number of global CAR-T clinical trials has dramatically increased, with that number more than threefolding over just the last 4 years from June 2016 to June 2020. And so while this is a wonderful activity for clinical treatment of patients with serious diseases, it also generates certain challenges. And in the case of lentiviral production, these challenges manifest themselves in terms of scalable and cost-effective solutions for generating the lentivirus necessary for transducing your cell types of interest. These current challenges lie primarily within the productivity of the systems. So low lentiviral titers will be difficult to deal with on the transduction end of the cell process. Scalability, and in particular with regard to adherent cultures, these cultures tend to be limited in terms of the volumes that can be produced, as well as safety and supply chain. So of the adherent culture processes, these utilize typically serum-containing media that has both safety and supply chain issues, as well as significant cost associated with it. So to address these problems with scalability and cost-effective lentiviral production, we have previously developed and launched the CTS LVMAX lentiviral production system. And this is a scalable, high-density, suspension-based system that generates high lentiviral titers, so in the neighborhood of 1 or greater than 1 times 10 to the 8 TU per mil, has xeno-free componentry. And in the CTS version of the LVMAX system, all of the reagents are manufactured under cGMP guidelines. So what is Gibco CTS? Maybe I'll just take a moment to dive into that. And Gibco CTS, or cell therapy systems, are GMP products that are designed to enable clinical and commercial cell and gene therapy manufacturing. So from a cGMP manufacturing standpoint, the reagents are manufactured in conformity with GMP for medical devices, 21 CFR Part 820, and following various USP and European Pharmacopoeia guidances. And these are manufactured at sites that are FDA registered and ISO certified and undergo regular audits. Similarly, the testing and documentation of the Gibco CTS reagents is enhanced compared to our research use only reagents. Documentation including drug master files, DMFs, and regulatory support files, as well as certificates of origin, are provided along with the reagents, as is relevant product safety testing, including such testing such as sterility, endotoxin, and mycoplasma on the relevant product classes. And the Gibco CTS products have proven use within FDA and EMA-approved CAR-T therapies, including the first FDA-approved cancer vaccine, and these products are cited in over 100 clinical trials. So just to give one example, one of the key components of the CTS LVMAX system is our viral production cells. And these cells are derived from HEC293 cells, and they've been suspension adapted into chemically defined medium and can grow to very high densities, so typically significantly greater than 10 million cells per mil in a standard shake flask culture system. These cells are free of the SV40 large T antigen and again are banked under cGMP guidelines. So on the right-hand side of the slide, you can see a number of testings that are completed for all of the CTS viral production cells. versus what is typically recorded for just RUO products, which more tends to be along the lines of assessing viability post-thaw of the cells, bacteriostatic and fungostatic activity, as well as mycoplasma testing. And on the CTS end of things, you can see that we've got at least 14 categories here that really cover the gamut of ensuring safety and quality of the cells that are going to be a vital part of your viral vector production process. So if we look at the components of the LVMAX system, what really makes the LVMAX system special is the way that all of these components have been developed to work synergistically with each other to allow for significantly enhanced titers of lentivirus. So again, starting with the viral production cells, a high-density suspension-adapted cell line that generates high titers of lentivirus. And this cell line has been mated with a chemically defined growth and production medium and supplement, as well as a robust high-efficiency transfection reagent and our lentivirus packaging plasmids. On top of this, a novel production enhancer is added to the cultures to further drive lentivirus production. And the results of all of these components working together synergistically is often 10 times or more lentivirus compared to comparable lentivirus production systems. All of these components are made to work together in a very streamlined production workflow that's very simple to be performed. And typically for the CTS LVMAX system, cells are prepared 1 day before transfection. So the cell stock would be seeded at about 3.5 million cells per mil. These cells are allowed to grow overnight, so they will overgrow that 4 times 10 to the 6 cells per mil requirement. And on day 0, we would take the higher density cells, add some fresh media to them, dilute them back to 4 E6 cells per mil, and then on top of that add 5% of the lentivirus LVMAX supplement to the cultures. The plasmid DNA for your lentivirus construct and packaging plasmids is diluted in CTS Optimem medium, and this is made up at 5% of the transfection volume of the culture. And similarly, the transfection reagent is then diluted in CTS Optimem, again at 5% of the volume of cells to be transfected. The diluted transfection reagent and diluted plasmid DNA are added together and complexed for 10 to 30 minutes, at which time they are added to transfect the cells, and followed by step 7, the addition of the enhancer. And then approximately 48 to 55 hours later tends to be an optimal time for harvesting lentivirus from the cell culture supernatant. And these improved titers through this LVMAX workflow can be seen when we compare them to more traditional processes that utilize PEI as the transfection reagent. So when comparing the LVMAX suspension system to an adherent PEI process, we can see on a volumetric basis about a 15-fold improvement in lentiviral titers. And compared to a PEI suspension-based process, about a 10-fold improvement in lentivirus titers. So all of these improvements in titer are also translatable to efficiencies in the downstream, as one would be processing significantly less culture supernatant in order to get the same amount of final viral titers. So taking into account the increases in titer as well as all other efficiencies of the LVMAX process, the LVMAX system can save over 50% of the cost compared to PEI-based lentiviral production methods. And the LVMAX system has also been demonstrated to be very scalable. And on the left, you see the scalability in terms of the lentivirus titer from a 96 deep well block plate, which typically would have a volume of about 1 to 1.2 mLs, to a 125 ml shake flask that typically has volumes of 30 to 50 mLs, all the way up to a 2-liter stir tank bioreactor ranging from about 600 mLs to 2 liters of production volume. And as you can see here, all of these titers are quite comparable, ranging from 1 ml to the 2-liter scale. And this scalability allows for the potential lentiviral production in the CTS LVMAX system to be significantly scaled up into even larger stir tank bioreactors. And what I'd like to do now is talk a bit about transferring the shake flask protocols for the LVMAX system into stir tank bioreactors. It's a fairly simple process. The cells and components are quite robust and easily adaptable over into the stir tank bioreactors. And what I'll do here is just go through the key steps of the process and then go into some data for an expression run in the 2-liter stir tank bioreactors. So here I'm giving an example of seeding the cells 3 days before transfection. This can also be cut down to 2 or even 1 days by simply seeding the cells at a higher cell density. But here we're showing that the cells are seeded at about 1 million cells per mil 3 days before transfection. And once again, the cells in step 2 will be overgrown compared to the 4 × 10⁶ target. So at day 0, at the time of transfection, cells will be diluted back with fresh media to 4 million cells per mil. And just as in the shake flask protocols, the plasma DNA is diluted into the CTS optimum medium, as well as the LVMAX transfection reagent. These are combined together in step 5 and added either by pumping into the reactor or by gravity feed to transfect the cells in step 6. And for simplicity in step 7, we've added the LVMAX enhancer and LVMAX supplement together and then pump them into the bioreactor as a single solution approximately 4 hours post-transfection. Once again, similar to the shake flask protocols, the lentivirus would be then harvested from the supernatant about 48 to 55 hours post-transfection. And if we look at on the left is a readout of some of the key conditions for the stir tank bioreactors for the LVMAX system. Here we had used a 3-liter high-performer glass bioreactor running off a G3 lab controller. Again, the seeding density can be from 0.5 to 1 × 10⁶ cells per mil or higher if you wanted to have a shorter duration until the cells were transfected. The final volume in this reactor approached about 2 liters once all the reagents were added together. DO was maintained at 40% through a macro drill hold sparge, with the stir speed at about 140 RPM, equating to, for this impeller, about 4.5 watts per meter cubed. Headspace air was at 50 mils per minute, and the pH of these cells for growth was found to be optimal at about 7.25, and this was controlled through CO2 only. Whereas the pH for transfection, the cells tend to express and produce lentivirus a bit better at a lower pH, so typically controlled at about 6.8 plus or minus 0.05 pH units. At the time of diluting the cells down to the starting transfection density of 4 million, the cells in this study were at about 6 million cells per mil, and the VCD at transfection, as mentioned, was about 4 million. DNA in this instance was used at 2.5 mg per liter of volume to transfect in a 3:2 packaging mix to transfer vector ratio, with the transfection reagent used at 6 mils per liter and the enhancer and the supplement added at 4% of the final volume respectively, 4 or 5% of the final volume respectively. And again, the enhancer and the supplement were added at 4 hours post-transfection, with the harvest about 48 to 55 hours. So in the top middle of the slide, you can see the viable cell density in the solid line and the cell viability in the dotted lines for the 125 ml control shake flask in the light blue line and the 3-liter stir tank in the dark blue line. So clearly it's evident to see that the profiles of growth for the cells in the shake flask and the stir tank reactor, both before and post-transfection, are quite comparable in either vessel, showing similar growth dynamics up to about 6 million or a little higher than that in the case of the stir tank reactor at day 0, where you can see the cutback to 4 million cells per mil, and then you can see the profiles take off from there over the next 48 plus hours during the lentivirus expression run. And on the right-hand side, you can see the comparability of the TU per mil as determined in an HT1080 infectivity assay, where the 3-liter stir tank is clearly equivalent to the 125 ml shake flask for the production of infectious lentivirus titer. So now I'd like to move into some of the downstream processing following the stir tank reactor work. And what I'd like to do here is give some tips and guidance around the key steps that are necessary for purifying lentivirus. Obviously, with lentivirus, speed becomes of the essence when purifying the virus. Elongated time in supernatant or hold times can diminish your infectivity of your lentivirus. So once started, it's imperative that one has a streamlined purification protocol in mind, keeping the time to an absolute minimum from the start of the process to your final harvesting and filtration and freeze down of your lentivirus. So step 8 is a continuation from the stir tank reactor slide. And here we're showing that once the 48 to 55 hours has passed, that the bulk harvest would have benzonase addition and magnesium chloride, typically approximately 2 hours at 37 C to allow for the digestion of any free nucleic acids in the bioreactor. And typically here is where we take our first samples for infectious titer and total lentiviral particles using, again, the HT1080 infectivity assay for infectious titer and a P24 ELISA for total lentivirus particles. And here it should be noted that we are working with a anti-CD19 CAR construct that has a V5 tag, and so in the HT1080 assay, infected cells are being monitored through a anti-V5 PE conjugated antibody by flow cytometry. And in each one of the steps, you'll note a couple of references for the steps that are described here. Many of these are quite prevalent in the literature, and again, it depends somewhat on your particular lentivirus and construct, but again, these are some general guidelines that are applicable for downstream lentivirus processing. So following the benzonase treatment, the supernatant is clarified by passing it through a depth filter. And this filtration train may start from a 5-micron pre-filter down to 0.8 or 0.45 micron final filtration into a suitable bioprocessing container. And again here, given some examples of different depth filters that have been used in the literature successfully for the clarification of the bulk harvest. Here, additional samples will tend to be taken, including residual host cell protein, which is monitored by a HEC293 ELISA assay, as well as residual host cell DNA that may be monitored by either qPCR or digital droplet PCR as a starting point to understand clearance in the later parts of the process. So now I will segue into step 10, chromatography, which is a critical step in the lentivirus production process. And here, quite typically, anion exchange resins are used with great success to purify lentivirus particles from the other components of the clarified supernatant. And here I show a number of examples, including the porous D50 resin, the SIMD and the SIMQ resins as well, that have been demonstrated to have excellent qualities for purifying lentivirus from your supernatant. In addition, there are other resins such as heparin affinity resins that have also been used for lentivirus purification, and these may again be dependent upon your process steps and your particular lentivirus and CAR constructs. Here, in the past, there had been some effort to use size exclusion chromatography to purify the lentivirus, but it's simply not efficient enough at removing the other process-related impurities, including host cell proteins, host cell DNA. So it is very important to move in the direction of either the anion exchange resins or the affinity resins to get that purity that you'll be looking for in your final product. So from here, the final UFDF and sterile filtration will take place typically through a TFF hollow fiber filter and a diafiltration step of 6 to 8 diafiltration volumes into, in this case, CTS Optimem medium, followed by your final filtration in a 0.2 micron filter. And again, just giving some examples of the typical analytics that will be performed at this step, although there are many others in addition to these that typically will be required to demonstrate product quality. And what we can see here, when we carried out this experiment, we actually ran our stir tank bioreactors with two different protocols. One that uses a more traditional PEI-based process, and the other our CTS LVMAX protocol. And a couple of major differences between these two processes. So in this instance, the PEI transfection took place at 2 million cells per mil, whereas the LVMAX transfection takes place at 4 million cells per mil to increase that biomass. And the difference here is that the CTS LVMAX, through the use of the supplement and the enhancer, is better able to handle the higher cell densities and therefore generating coordinately higher lentivirus final titers. What we see here is that at the time of harvest, the CTS LVMAX system was at about 3.69 million cells per mil, versus the PEI process at about 1.7 million cells per mil. And it should be noted that in this instance, the cells and the medium were consistent between the PEI process and the CTS LVMAX process. And again, at the time of harvest, viability was quite similar at about 60 to 65% between the two processes. And what you can see here is that through the robust downstream process that I described previously, the host cell protein, even though it shows to be somewhat higher early on in the clarified culture for the CTS LVMAX system, that's not unexpected given the double cell density of the system at the time of transfection. But what it clearly demonstrates is the downstream process through the anion exchange resin step is clearly able to take care of the host cell protein and reduce it to levels that are comparable and in fact were even slightly lower than the PEI-based process. So we had no issues there handling the additional host cell protein through the anion exchange and purification process that I mentioned in the previous slide. On the right-hand side, you can see the final titer for lentivirus that was filtered through the 0.2 micron filter. And indeed, we had about a 6-fold higher final lentiviral titer using the CTS LVMAX system compared to the PEI process. And in fact, overall, we had about 10% higher final lentiviral recovery for the CTS LVMAX system versus the PEI process. So again, with the downstream processing steps mentioned in the previous slide, we had no problem recovering our LVMAX-derived lentivirus, and in fact had slightly better recovery percentages compared to the PEI process. And lastly, to ensure the infectivity of our CD19 CAR lentiviruses, we activated pbmcs for 72 hours with anti-CD3, anti-CD28 in Vitrogen Dynabeads, and we transduced these activated pbmcs with our lentivirus, cultured them for approximately 3 days, and then we assessed the anti-CD19 CAR T cells that were a result of this infection by flow cytometry. And on the bottom left, what you can see is the flow cytometry analysis where we were gating for these CAR T expressing T cells using the anti-V5 antibody, as I mentioned previously. And on the left, you can see the negative control non-infected T cells, a very clean population with essentially no cells showing positive with the anti-V5 tag. And on the right-hand side, from the LVMAX system, showing about 11.6% of the cells that are positive for the CD19 CAR construct. And if we look to the right, we look at the T cell transduction efficiency, using a relatively low MOI of 2 to make it easily decipherable differences in the lentivirus between the PEI process and the LVMAX process. And we can see that, again, the LVMAX had about an 11% transduction efficiency versus closer to about 2% transduction efficiency for the final PEI filtered product. So clearly here, we're able to take the LVMAX system from a stir tank bioreactor, put it through a robust downstream process to reduce host cell protein, host cell DNA, and still have good recovery of our lentivirus at levels that are comparable with more traditional PEI methods. We are able to obtain significantly higher titers, and the lentivirus from this process is able to transduce T cells and generate these anti-CD19 CAR T cells. So in conclusion, the CTS LVMAX lentiviral production system provides a streamlined solution for the production of high-titer lentiviral vectors in very scalable suspension cultures. Excellent scalability of lentiviral titers can be observed from the milliliter to the multiliter scale in a variety of different formats, including multi-well plates, shake flasks, and stir tank bioreactors. Multifold improvements in the final lentiviral titers were achieved in the LVMAX system compared to a PEI-based process, and the downstream purification processes were able to reduce host cell protein levels and provide final lentiviral recoveries equivalent to or greater to those of the paired PEI-based process without any modifications between the LVMAX system and the PEI-based processes. And finally, the resultant purified lentivirus from the CTS LVMAX system was capable of transducing human T cells in vitro using a relevant anti-CD9 CAR construct. And with that, I would like to thank you for your time, and we look forward to answering any questions that you may have. Thank you very much.
Okay, great. Thanks, John. So the first question is, what are some of the best practices when establishing the LVMAX system in our lab the first time to ensure maximum lentiviral titers?
Yeah, thank you, Leah. It's a great question, and this question is really important to ensure a baseline for your lentiviral titers going from small scale to larger scale. And this really is, a lot of these best practices are consistent, whether we're talking about making protein or lentivirus or AAV, for example, and they revolve primarily around ensuring that you have optimal cell culture conditions. So in any of these systems, your cells are the foundation for the system, and ensuring that those cells are handled according to the instructions provided in the manuals is critical. And that comes down to keeping a close eye on the density of the cells at the time of subculturing to ensure that they're either not undergrown at the time of subculturing. So high density cells, such as the viral production cells of the LVMAX system, they prefer to be at least in certainly early log phase growth before they are subcultured. If they don't have a chance to attain that and they are oversplit multiple times, their cell growth characteristics can become perturbed. Similarly, you don't want to overgrow your cells too much where you're getting into late log phase or actually closer to peak cell density. So important to keep the cells somewhere in that 3 to about 5 million cell per mil range at the time of subculturing. Similarly, it's important and really critical to match your shake flasks, for example, with the appropriate volume of cell culture media as well as your speed and orbit on the shakers. So typically, for example, for a 125 ml flask, one would use about 30 mLs of culture volume and typically about 125 RPM on a 19 millimeter shaker, a little bit lower for a shaker with a larger orbital diameter. So all of these things are really critical in order to ensure cell health, and at the time of transfection, really making sure that your cells are approaching 95+% viability so that you have very healthy cultures at the time of transfection. And a lot of these best practices, again, will be critical to allow you to understand the growth characteristics of the cells, doubling times, VCDs, etc., and ensure that those can be ported over when you go to troubleshoot and optimize at the larger expression volumes.
Okay. So for the incubation time of 10 to 30 minutes for transfection, can you suggest the ideal in this time frame window? Is it better to sit closer to 10 minutes or longer towards the 30 minutes?
So that's a great question, and typically you want to have about 10 to 12 minutes at the bottom end of the complexation to allow for full complexation to take place. And then we typically build in about that additional 20 minutes in order to, for example, if you're doing this in a larger vessel or a bag, to be able to then either by gravity feed or pumping into the bioreactors, for example, you have about another 15 to 20 minute window after that initial 10-minute complexation takes place to get the complexation into your vessel, again in the case of the larger vessels. So really anywhere within that window is fine if you're at the small scale. Sticking to 10 to 15 minutes should be fine just to save a little bit of time, but that additional complexation stability up to 30 minutes gives you the flexibility needed to get into the larger vessels.
Okay. Can LVMAX be used for AAV?
So the LVMAX system, we can provide optimization guidance for making AAV in the LVMAX system. And in fact, that's not terribly uncommon today because we do offer the LVMAX system in the CTS version, so fully manufactured under GMP compliance. So we do have a fair number of users that are making AAV in the LVMAX system, even with plans into the clinical production. So we are working on our AAV Max system for launch in the 2021 timeframe, which will be fully optimized for the production of AAV. But certainly we can be contacted and provide support and protocols for those that are looking for a solution today for using the LVMAX system to produce AAV.
Okay, I think I'm going to paraphrase this question, but can you please advise us how to minimize cytotoxicity?
So again, I think it goes back to what I mentioned, that cytotoxicity, the first aspect is that you always want to make sure that your cell culture conditions are optimized. And that, goes for really, any construct that you would be expressing. The first step is that you have healthy cells that are growing well within their log phase, that are very high viability at the time of transfection. And then the second step in terms of toxicity also revolves around ensuring that your plasmid ratios and your complexation ratio between your transfection reagent and your plasmids is optimal, such that you're not putting undue stress on the cells. And typically with our VeraPower LV packaging mix and the guidance provided in the protocol, you can be relatively assured that within that mix for non-toxic constructs, that the ratios provided would be a very good starting place. However, certainly there are constructs that can be toxic to the cells. And in those instances, other strategies that may be more in depth may need to take place. So I think it depends, primarily upon whether the toxicity is coming from your conditions and whether they could be further optimized, or if it's the construct itself that's leading to toxicity. And again, in that case, it's a more difficult question to address because it may be much more specific based on that gene of interest, rather than the upfront optimization to ensure that your cell culture conditions and your shaking or bioreactor conditions are optimal, so that cells are not being having undue stress placed on them and unable to stay viable throughout the production run.
Okay, have you done large-scale productions of 50 liters or greater, and if so, were there any issues with the robustness of the transfection reagent, and how did you deal with it?
Mm-hmm. So that's a great question. So the data that we've shown today for the 2-liter stir tank bioreactors is sort of phase one. So we'll be coming back with additional installments as we continue to work through all of the analytics and the downstream optimization for the 2 to 3 liter bioreactors, with the intent that we will be moving up to 50-liter bioreactors this year. So I can say that certainly outside of the work that we're doing, 50-liter bioreactor runs for the LVMAX system have been performed and have been performed successfully. with the cells scaling up very well from the 2 to 10 or 15 liter to the 50 liter reactions. And in general, we've not seen any issues with the complexation for the larger volumes. I think typically, again, with any transfection reagent, you want to keep in mind the timing of the complexation, and most typically, if this is done in a bag or bottle, the componentry is added together, swirled or mixed gently, and just allowed to incubate for that period of time before pumping it into the reactors. So again, there may be some differences there, whether or not we're looking at, gravity feed or pump feeding, tube diameter, speeds, etc. But those are also, items that we would be more than happy to provide our guidance on as well. But to date, we've seen, no red flags in terms of any aspect of the system getting to the 50-liter scale. And again, we expect to be able to provide direct updates on that as the year progresses.
Okay, so it looks like we have time for just one more question. All the questions will be passed directly to John, and he can follow up with you individually after the webcast. So go ahead and keep typing your questions in. If you have more questions, we'll pass those along. But the last question then is, why use Optimum to dilute plasmid and transfection reagents? Can other media be used for the dilutions instead?
Yeah, that's a great question, and it's really interesting. Sort of the history of the use of Optimum for transfection is just that it's been a good, robust complexation media that works well with many different transfection reagents, and so it's often fallen back upon as the complexation buffer of choice, for different transfection reagents. But certainly other medias can be used. You tend to have to be a little bit cautious when you start getting to the higher density medias that are more rich in nutrients to allow for higher density growth of the 293 cells. So, for example, taking the LVMAX media, complexing into that, is it possible? Absolutely, yes. Will you likely see a reduction in titers compared to Optimum? You probably will see a slight reduction compared to Optimum. I think I would just keep in mind the notion of, the leaner medias tend to do a pretty good job of complexing the DNA, but it's something that, you would certainly want to test in your system. And again, Optimum really has been chosen simply because it's well known to be a robust and good middle of the road complexation buffer, with minimal downsides to its formulation.
Okay, great. Thank you, John.
Thank you.
And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive an email providing you with a direct link. We look forward to having you join us at our future BioProcess International Ask the Expert webcast. Look for those announcements in your inbox. Goodbye.